Now, the major question for OT is, are there, because 6 out of 13 patients responded on the phase 1, we'll see sort of how it pans out with larger studies, but that clearly means that not all patients are going to respond to OT post-relapse. And what's truly lacking in small cell lung cancer that's revolutionized other lung cancers or other cancers in general is biomarker-directed therapy. So we wanted to find, we have this system, it's matching what happens to the patients. We have a ton of models and anchor points from models from patients on the trial to say, well, this response in a mouse mimics a resistant or a relapse, and this response mimics a sensitive. We can use this system to try to identify biomarkers for OT, and that's what the co-clinical trial was. It's literally occurring as the clinical trial is occurring in order to optimize the next clinical trial or application of it using these models. And we found, we're in the middle actually of the co-clinical trial. We're still looking for better biomarkers and trying to find out some of the mechanisms of resistance for mechanisms for the biomarkers that we've already found. But early on we found that one gene of interest called SCLC11, which other groups have sort of looked at, in our system is matching up with sensitivity to Olaparib temozolomide, and we were able to sort of validate that finding in our system, and I think there's interest in using that as a biomarker for application of PARP inhibitors in small cell. And then also we found some new ones. We found one of them which would be very difficult to find by sort of massive genomics or proteomics, because it's actually not a protein. It's the enzymatic effect of PARP1. It's called PARylation. So PARP1, what it is, it adds on polyadenosine ribose groups to other proteins. There's a good antibody for this, so we can see it. And what we ended up finding was that although PARP1 level itself doesn't really match up with whether the model or potentially the patient sensitive or resistant, the level of PARP1 activity is measured by PARylation without treatment. That does seem to match up. So the more active it is, the more sensitive the model is, and that's the major findings so far from the co-clinical trial, but stay tuned.